All right, let's jump right in. Today, we're tackling a big one, human alphaherpesvirus 2, better known to all of us as Herpes Simplex Virus 2. Now, this is a pathogen with some serious, lifelong consequences for patients, which makes it a constant challenge in our field. We've tailored this explainer specifically as a briefing for you, our fellow Dutch medical microbiologists, hitting the key details you really need to know. Just a quick note on our sources here. All the information you're about to see was pulled together using a Gemini deep search. And if you really want to get into the nitty-gritty of the primary literature, the complete source list is waiting for you in the linked Notebook LM document. So to really set the stage, just think about the sheer scale of this virus. It's literally everywhere. A diagnosis is, in many ways, for life. And the clinical headaches it causes? They pop up all over the place, from a standard outpatient visit all the way to the high stakes of the neonatal ICU. That right there is exactly why a crystal clear understanding is so critical for us. Okay, so first things first, let's get back to basics and profile the virus itself. Getting a grip on its structure and how we classify it is really the foundation for understanding everything else it does in the clinic. So you see its formal classification here, slotting it right into the alphaherpesviridae subfamily. Now that is the key piece of the puzzle. Why? Because this entire group is famous, or maybe infamous is a better word, for setting up shop for life. They establish these latent infections in sensory neurons, and that is the absolute heart of its entire disease process. So, what does this thing actually look like? Well, the virion is this really complex, multi-layered particle, kind of like a Russian nesting doll. You've got the DNA core, wrapped in an icosahedral capsid, then a protein layer called the tegument, and finally a lipid envelope on the outside. Sticking out of that envelope are these crucial glycoproteins, and for us in the lab, there's one we really care about, glycoprotein G, or GG2. Its unique shape is exactly what our type-specific serology tests use to tell the difference between Herpes Simplex Virus 2 and its cousin, Herpes Simplex Virus 1. It's our molecular fingerprint. All right, so we've got the blueprint of the virus. How does that actually play out in a patient? Well, this virus is responsible for a huge range of diseases. It runs the gamut from relatively common problems to infections that are just absolutely devastating. Now, of course, everyone thinks of genital herpes, and for good reason. It's the classic presentation. But that is not the whole story. Herpes simplex virus 2 is also a major culprit behind aseptic, recurrent lymphocytic meningitis. And then you get to the really scary stuff. At the most severe end, we're talking about two bona fide medical emergencies, herpes simplex encephalitis and neonatal herpes. With these, there is no time to waste. You need a diagnosis, and you need to intervene immediately to have any hope of preventing a catastrophic outcome. Which, of course, brings us right to our turf, the lab. Let's talk diagnostics. And honestly, the story of diagnostics here hasn't just been an evolution. It has been a total revolution. We've seen a massive paradigm shift, moving away from the old school methods to the molecular gold standard that we all rely on today. And this comparison really tells the whole story, doesn't it? On one side, you've got viral culture, the old standard, but let's be real, its sensitivity is just not good enough, especially for healing or recurrent lesions where it can drop below 50%. Plus, it's just slow. On the other side, you've got NATs, or PCR. It's a completely different ballgame. They are incredibly sensitive. They can pick up tiny amounts of viral DNA, and they're fast. That's why they are, without a doubt, the gold standard for any active infection, and especially for anything involving the central nervous system. So let's lay out all our options here. The main takeaway is this. NAT is king for diagnosis, no question. But, and this is a big but, viral culture still has one absolutely critical job, providing us with a live isolate for susceptibility testing. And I want to draw your attention to that bottom row, because this is a message we need to hammer home. IgM serology, it is not type-specific. It is clinically useless. Just don't use it. End of story. All right, let's shift gears from the lab to the clinic. It's time to talk about management. We're going to walk through the Dutch guidelines, see how they stack up against the international ones, tackle the thorny issue of resistance, and then focus on a few really important patient groups. So here in the Netherlands, the NHG guidelines for genital herpes are pretty straightforward. For that initial first episode, the go-to is valacyclovir, 500 milligrams, twice a day for 5 days. And if a patient has a reoccurrence and you're treating it episodically, it's the exact same regimen. Okay, but now let's see how that compares internationally. For instance, with the CDC guidelines. Right away, you see a pretty big difference. For that same first episode, the CDC goes for a much higher dose, a full 1,000 milligrams twice a day, and they recommend treating for longer, 7 to 10 days. This isn't just a trivial point. It's a really important distinction to have in your back pocket when you're reading international studies or managing patients. Now, when it comes to high-risk groups, thankfully, there's pretty solid agreement across the board. In pregnancy, the name of the game is preventing neonatal herpes, period. That means starting suppressive therapy at 36 weeks. And if you have a neonate with even a suspicion of herpes, that is an all-hands-on-deck medical emergency. They need immediate hospitalization and high-dose IV acyclovir. And then, of course, our immunocompromised patients. They often get hit harder, and this is the group where the alarm bells for antiviral resistance really start ringing loudest. So we've mentioned resistance, but what's actually happening? How does the virus pull this off on a molecular level? Well, there are two main ways the virus outsmarts the drugs. The most common one, by a long shot, involves a mutation in the viral thymidine kinase gene, or TK gene, that's UL23. See, acyclovir is a prodrug. It needs the virus's own TK enzyme to flick the on switch. If that enzyme is broken because of a mutation, the drug just sits there, completely useless. The other, less common mechanism is a mutation in the DNA polymerase gene itself, UL30, which is the ultimate target of the drug. And when I say the TK mechanism is more common, I mean it is overwhelmingly more common. Think about this. More than 90% of all the resistant Herpes Simplex Virus 2 strains we find in the clinic have mutations in that thymidine kinase gene. That's exactly why when we do perform genotypic resistance testing, we're almost always focused on that specific pathway. And this brings us full circle, right back to a vital point for the lab. To actually confirm resistance and guide second-line therapy, we need to do phenotypic testing. And for that, we absolutely need a live virus isolate from cell culture, typically grown on lines like A549 or Vero cells, until we see that classic cytopathic effect. This is the reason why viral culture, despite its diagnostic limitations, remains indispensable whenever we suspect treatment failure. Okay, to finish up, let's talk about prevention and control. What are the practical, real-world steps we can take to stop this virus from spreading? Look, there's no single magic bullet here. Preventing transmission requires a multi-pronged attack. You've got the behavioral side of things, consistent condom use, and avoiding contact during outbreaks. Then you have medical interventions like suppressive therapy, which is a game changer because it cuts down on recurrences and that tricky asymptomatic shedding. And you wrap all of that up with good counseling for partners and those strict protocols for pregnancy. It's the whole package that makes a difference. Which leaves us with the big million-dollar question, doesn't it? When you're dealing with a virus that hides out for life in your nerve cells and can spread even without symptoms, what does winning even look like? What is the ultimate endgame for controlling human alpha herpes virus 2? It really forces us to look past just managing symptoms and start thinking bigger about what's on the horizon, like therapeutic vaccines, and maybe, just maybe, an actual cure one day. And that's a wrap for our deep dive on human alpha herpes virus 2. We really hope this briefing was useful for you. If it was, please do us a favor and subscribe for more focused explainers like this one. And hey, drop any questions or feedback you have in the comments below. We'd love to hear from you. Thanks for joining.